participate by having a poster presentation, and then there is a competition, so the best posters will be given awards. Okay, this is not only at the level of the College of Medicine, but also at the level of the university. So students from all colleges will be participating in this poster research day. Any other questions? I put 1 minute.
My Wi-Fi was
Okay, come to me at the end. Okay, come to me at the end. You did the attendance didn't work? Who had an issue with the attendance? Raise your hand if you had an issue. Only two. Talk to me at the end. Yes, what was your question, please? No, we have this research day every year, okay? For the last several months. It's not the first time. Any other questions? Okay. By the way, there will be changes in the CVP schedule as it is posted now. First, to reflect the fact that now the year is shortened because you have to finish your exams before the 21 of Ramadan. So there will be revision in the CVP schedule based on that. And also, I think on the 3rd of March, there is the progress test. So that the test that is required for all the years. So you will be sitting for this test too. So it will be on the 3rd of March. So there will be some rescheduling. So always keep checking your schedule. It's done for all medical schools in Saudi Arabia, and they compare how the level of the students change every year. So basically, all years you take the exam, and then they compare the performance of all the years, and then they compare it to other medical schools. It's an important test for you to attend because I think it's also similar to licensing exams, etc. So it's a good practice for you, and you will not have classes, so you have to attend. Anyway, they will give you an introduction about it near the time. Okay, how many of you have done or heard about PCR? How many of you know what is a PCR reaction? Okay, good. So all of you did, raise your hand, you did A level? Yes. No? How do you know about PCR?
I took it in high school.
You took it in high school, so international school? Okay. In Saudi school, did they tell you about PCR? What's PCR in Arabic, by the way? PCR? Anyway, so don't worry, even if you don't know anything about PCR, I will be introducing those stuff to you today, so don't worry, even if you don't have any previous knowledge. Okay, so the way I summarize the techniques in this PowerPoint is that I will talk about techniques to study DNA, then techniques to study RNA, then techniques to study protein. Okay? So this is how I group them. I advise you to read the book, the last chapter in your Lippincott book. This is where I took most of the material. It will not be confusing because most of the information I'm presenting here is actually taken from the book. Okay, so the last chapter in your Lippincott biochemistry book, this is where I took the information for these slides. Okay, so make sure you read the book. Right, we will start by talking about the techniques for DNA. Okay, so we will start about talking. We'll start by talking about the techniques that used for isolating and studying DNA. Okay, so we're going to talk about things like PCR, blotting techniques, DNA cloning, restriction enzymes, gel electrophoresis, etc. So most of these techniques you will be able to do in your experiment, but the others, they are all related. So by attending the lab and participating carefully in the lab, you will have really a good idea about the molecular techniques. Also, I strongly recommend for you to open all the animations and the videos, which I have embedded in the PowerPoints, because it's very important for you to see those techniques in, when they are being done in the lab, rather than just reading the theory. Okay, so please do watch the videos. Okay, so a big advantage in recent days in studying DNA was the discovery of three things: the discovery of restriction enzymes, the discovery of DNA cloning procedure, and the probes. So these three things are used in different techniques, as we will see today. But you need to know that molecular biology is a relatively new field. Actually, PCR started in the 1980s, so this is where people started to amplify DNA, and since then it has been a revolution of molecular biology. Being able to amplify DNA and study DNA is something new, which previously people were not able to do. Okay, so what are restriction enzymes? So restriction, what does it mean? What does the word restriction mean? Yes, restriction here doesn't mean like you restrict something, you cut it, okay?
Yeah.
So restriction endonucleases. What does the word endonucleases mean? So they are enzymes, so nucleases, they are enzymes that digest what? DNA, nucleic acid, right? And endo means they cut it within. Okay, so basically restriction endonucleases are enzymes that will cut DNA sequence inside the DNA. They will make specific cuts inside the DNA. Okay? So how would they cut any DNA or specific DNA sequence? How do they recognize which DNA to cut? It all has to do with the restriction sites, or the what we call the palindromic sequence. Okay, so I'll show you here. So a restriction enzyme will recognize a specific sequence, and this sequence is about 6 base pair, okay? It will read that sequence and will make a specific cut. So each enzyme has a specific restriction site. So we have a restriction site for each restriction enzyme. And the way this sequence is arranged, we call it a palindromic sequence. What does palindrome mean in English? Yes?
Like river, you can read it from backwards.
Yes, so a word that is palindromic, you can read it the same way from right to left, left to right. In DNA, we don't have right and left, we have 5' and 3'. So if you look at the sequence here, if you look at the top sequence, can you read it from 5 to 3? What does it read? Okay, now let's look at the bottom strand and try to read it 5 to 3. Okay, so it reads the same, right? The top strand and the bottom strand, they read the same from 5 to 3. So this is a palindromic sequence. So this is a typical example of a sequence that is recognized by a restriction enzyme. So the enzyme, once it sees that sequence, it will make a specific cut. Okay, so it has to read that sequence in order to cut it. Okay, so these enzymes, where do they come from? Where do we get them from? They come from bacteria, yes. So this is a list of the enzymes. I'm not sure, can you see the names? Yes? No? Okay, so the name of the enzyme could be something like EcoR1, BAMH1, HIND2, or HIND3. So what are these names? They are all given based on the name of the bacteria from which they are isolated. So restriction enzymes are isolated from bacteria. They are enzymes present in bacteria that scientists isolate, and then they use them to cleave or cut DNA. Okay? So that's why they have different names based on the bacteria from which they are isolated. Do we really need to know the naming? How do we name them? No, because you just need to have an idea that they come from the bacteria. You'll be taking POD next year and you will know more from microbiology, but for now just know that Like HIND3 is isolated from the Haemophilus influenza bacteria. Okay, what is the Roman number? So we have 1, 2, 3. What does the Roman number mean? It's the number of the enzyme. Like if they have isolated more than one enzyme from the same bacteria, so the first enzyme to be isolated will be given the Roman number 1, the second 2, and so on and so forth. Okay? So these are the restriction enzymes. These are just examples. Of course, please don't ask me, do we need to memorize this? Because it means that you have no clue about molecular biology. So these are the restriction enzymes, and these are the restriction sites which are recognized and where the enzyme is going to be cut is indicated by arrows. Okay? And this is the cleavage product. So this is the resulting product. Okay? But of course you have to think that this is just a fragment, but there will be DNA here and DNA here, so it will cut at a specific sequence producing different size fragment. This is very important for studying mutation. So if you want to study mutation or polymorphism, a restriction site can be modified by a mutation, okay? So a mutation could either introduce a new restriction site, or it could abolish a restriction site, okay? So by cutting the same DNA, whether it's from a person with a mutation or a person without the mutation, you can compare those DNA fragments, and you will be able to say whether this patient had the mutation or no. And we will come back to this later. Okay? So this is restriction enzymes. Enzymes isolated from bacteria. We use them to cut DNA. Do we use restriction enzymes on their own as a technique? They don't stand as a technique on their own. We might use them after a PCR, or we might use them for cloning. So it is always part of a procedure that you're doing in the lab. Please watch the animation. The next technique, which is very related to the restriction enzyme use, is DNA cloning. Who can explain to me what's DNA cloning? In brief. So when we are doing cloning, we're trying to make a copy. We're trying to make multiple copies of the same thing. Okay? So when you clone a sheep, yes, you want to make one copy of the same sheep. But DNA cloning, when you try to make multiple exact copies from one piece of DNA, yes?
Yeah, so basically you cut out the DNA the way you said, and you introduce it into a bacteria, but it's a plasmid, and then the bacteria will then produce the protein you want. Then you take the protein that you want, and then use it whatever.
Yes, well done, thank you. So basically, it's trying to make multiple identical copies of a DNA, and they use it in pharmaceutical company to clone proteins. Okay, so like this is how they make insulin. Okay, so they make what we call recombinant insulin, or this is how they make growth hormones that they give to people who have stunted growth. So if they want to, help these children to have a better growth, they might be given growth hormones. So how do they make them in the lab? They take the human gene, let's say for the growth hormone, okay, and then they insert it into a bacterial plasmid. You know what is a plasmid, right? Yes. Okay, the extra chromosomal DNA of the bacteria. They insert that human gene inside the plasmid, and this is what we call recombinant DNA, because they are making a new DNA that doesn't exist in nature, okay? And why do they put it in the plasmid of the bacteria? What is the purpose of inserting it in the bacteria? Yes, because once they return that plasmid back to the bacteria will multiply, and as the bacteria will multiply, the plasmid will multiply, and they can have millions of copies of that DNA, okay, which was replicated inside the bacteria. So they use the bacteria as a host to grow multiple copies of the human gene in order to make tons of proteins from it. Okay, so it's like they're using the bacteria as a factory to make many of this protein. Okay, so this is DNA cloning in brief without going into detail. Okay, so it's one way of amplifying DNA. Another way of amplifying DNA would be what? PCR, and we are going to talk about it later. Okay? So this is DNA cloning. Of course, you need restriction enzymes when you are doing DNA cloning. Why you need restriction enzymes?
To prevent mutations.
To remove the...
To cut the DNA, because this is the plasmid DNA, it's circular, right? You need to cut it first. So here they will cut it with the restriction enzymes, for example, EcoR1, and they cut the DNA of interest with EcoR1 also in order to make it sticky so that it can stick here, because they have to have complementary and in order to stick together, okay? So they will cut the plasmid DNA with the same enzyme that they use to cut the gene of interest in order for the ends of these two DNA stick together to form the recombinant DNA. Clear? Okay, so this is from your book illustrating the technique, how you insert a foreign DNA into a plasmid DNA, then you let the bacteria multiply, and then you can use that DNA for research purposes, or you can use it to make multiple copies of a protein. Okay, so this is regarding the use of DNA cloning. Okay, so you can use it in medicine, but also you can use it in environmental, industry and agriculture.
Doctor,
Yes.
when they use restriction enzymes, they have to have their own cleavage site in the plasmid, right?
Yes.
How did they find it?
Once you order, this is on the second slide. So once you order, if you're doing research or you're doing an experiment, whatever it is, so if you purchase a certain, they call it plasmid or vector because it's going to take the DNA. So if you purchase the plasmid, they will send you the plasmid in a tube and they send you a map with it. So you know what is the sequence here, and you will know which enzyme will cleave that plasmid so that you can use them. So here they give you a choice. Usually they don't give you one enzyme. You have to have multiple choice because you need to have that restriction site also in your gene that you are going to insert. So you can see here we have 5 options of restriction enzymes, okay? And also they will give you an idea because these plasmids usually are engineered so that they have antibiotic resistance. Okay, so this plasmid have antibiotic resistance to tetracycline and to ampicillin. Why you need antibiotic resistance? So that when you are doing the experiment, you only grow bacteria which carry the plasmid. Okay, so only the bacteria that have the plasmid and therefore have the resistance to this antibiotic will grow. So when you do this in POD next year, when you grow bacteria on a petri dish, usually you put antibiotic in order to prevent any other bacteria to grow because the petri dish is like a medium for growth for anything. So usually you have to add an antibiotic so that only certain bacteria will grow, and in this case, only the bacteria with the resistance gene will grow. Clear?
So, doctor, how can they choose a specific bacteria that have a resistance to this antibiotic?
No. The bacteria will have in it this plasmid, okay? And therefore, only the bacteria which has inside this plasmid will grow.
So the resistance is still to the plasmid?
To the plasmid, okay? Clear? Okay, let's move on to the next technique. It is gel electrophoresis. So this is explained in here. So basically, what are we doing when we are doing gel electrophoresis for DNA? We're trying to visualize DNA. Okay, because DNA, you've done DNA extraction last term. Were you able to see the DNA? No. So in order to see the DNA, you need to load it on a gel. What does the gel mean? Gel بنحطه على شعرنا? No. You need to prepare a gel in the lab by using a material called agarose. Okay? So how do you prepare this gel?
Microwave.
The same way you prepare Jello at home. So there is a powder that you will mix with a solution. لسه ما بلش الفصل. We just started the semester. We should be all enthusiastic and keen on getting A's and paying attention and waiting for me to say the word so that you grasp it, not chatting to your friend. Okay, so you prepare the gel by mixing agarose with a buffer. You pour it in not in a plate, you're not going to eat it, so you pour it in a tray, and this tray you will add to it a comb. The comb, similar to the comb you brush your hair, it is something, a plastic that has in it teeth, and the purpose of using this teeth is to make what? To make these holes. What do we call these holes in molecular biology? Well, yes. So you are making wells, and in these wells, so for too much terminology, that's why you need to study this before you go to the lab. So we said we make agarose gel electrophoresis. Okay, so you need to use the powder agarose, mix it with the buffer, use a comb that has teeth in it, and the teeth will make the wells. Why you need wells? Because you want to load, okay? Not upload. Somebody told me upload. You don't upload the sample. You load the sample using a pipette into the well. Why into the well?
To store it.
No, yes, because you want the sample to travel inside the gel. Okay, so what we are trying to do is to put DNA inside the agarose. So how DNA will go inside the gel? By going first into the well, and then from the well, the sample will travel inside the gel. What will make the DNA move? The electricity. So we have to take the gel, put it into an electrical instrument. So you will have a cathode and anode, and the sample, the DNA has which charge, negative or positive?
Negative.
Negative. So the sample will migrate toward which end?
Positive.
It will migrate toward the positive. So we are separating DNA according to the charge and also according to the size. Okay? So which DNA fragment will move faster? The shortest. Okay? So the shorter fragment will move faster. Okay? So that's what you are trying to do. Separate DNA on a gel according to the size in order to see the DNA and to see whether it is the right size or not. Okay? So how do I know the size? Do I use a ruler to know the size? No, I have to compare it against this. What do we call it? We can call it a standard, a marker, or a ladder. Okay, they all mean the same thing. So it's basically a sample with fragments of known sizes. Okay, so that when you compare them, let's say, this, I know that this is 2,000 base pair, this is 1,000, this is 1,200. So by comparing my samples, so each one of these is the well. So I had loaded my sample here, and the sample has traveled in this direction. Okay? So by comparing my sample fragment size to the ladder, I will be able to give an estimate of the size of my samples. And each one of these is a well with a sample loaded in it. So each one of these will be one of those. Okay? This is how you see it in naked eye. No, you will not see the bands in naked eye. In naked eye, you just see some blue color, which is caused by the loading buffer. So when you load the sample, because DNA is colorless, when you load the sample, you are going to add to it something like methylene blue, so that you can see the color as you load the sample in the well, because you're going to be loading something like 2 microliter, and you want to be able to see whether the sample is going in the well or it's just floating. So that's why we need to have a loading dye. But it's not the loading dye that will result in this color. How we will be able to see this color, this red color? Because it's written in the note somewhere here. We are going to use a stain, and this stain is called ethidium bromide. Okay? So what is ethidium bromide? It's a stain that will bind to DNA and will give it this red color, not under normal light, but under UV light. You need to use ultraviolet light. So there will be in the lab an ultraviolet lamp, and you will put your gel on it, but it has to have a screen to prevent your eyes from damage, because as you know, ultraviolet light, you should not see ultraviolet light with your naked eye. So you will be able to see those bands of DNA after you had stained with ethidium bromide and after putting the UV lamp on. Okay? So which band is larger in size, the top one or the bottom one?
The top one.
The top is the larger in size. So these are the shortest fragments.
So the shorter it is,
The faster it will migrate, because it's traveling inside the gel. The electricity is making it move, but its size is determining how fast it's going. So the larger it is, the slower it will move, because it's facing resistance inside this porous gel of agarose. Okay? Yes.
Every pink object is one strand.
It is, no. Every pink object is one band of DNA. Okay?
Double strand.
Double strand. Okay? So this is how you see the DNA on a gel. Clear?
Yes.
Any questions regarding gel electrophoresis? So in the lab, you will be able to do PCR, we'll talk about it in a minute. You will be able to load your gels. So you're going to practice doing this in the lab. So because of the time, they will prepare the gel for you, so you will not pour the gel. The gel will be already prepared for you. So you will practice loading the sample, and then you will see the gel, you will see something like this in the lab. Okay? So this is gel electrophoresis that is used for visualizing and determining the size of DNA. So we don't do it on its own. So usually you do it after a PCR, or you could have done it after DNA extraction. So you load the sample on a gel in order to see it and determine whether you have the right thing or not. Okay, the last topic that I will talk about today is PCR, or polymerase chain reaction. Okay, so what is the purpose of doing PCR?
Get samples of DNA.
Yeah, so basically you're trying to amplify DNA. Okay, so we want to amplify a gene. Let's say I want to amplify the beta globin gene from the DNA of the blood of a patient to see if they have sickle cell or no. So what do I need to make a PCR? So I'm trying to amplify the DNA. I will need to amplify the DNA using a PCR machine, okay, which is also called the thermal cycler. So the machine will look something like this. So in this machine, I will put the test tubes, and in the test tube, I will have my mixture of the DNA that I want to amplify. And the machine will expose the sample to cycles of different temperature. Okay? So why we need those different temperatures? So basically what we're trying to do, first, we need to heat the sample so that we denature the DNA. So we need to separate the double-stranded DNA. Okay, so the first heating is to denature the DNA. So usually we heat to 90 degrees to denature the DNA. Then we cool to something like 50 degrees or 56 degrees, depending on the primers that I'm using. So what are these primers?
Oh, tap memories.
Sorry?
Tap memories, something.
No, the primers, they are the short sequence of DNA that you have designed in order to amplify a certain gene. So if I want to amplify the beta globin gene, I need to amplify primers for the beta globin. So these are short sequence of DNA here in green, flanking my gene of interest from each side. And what they will do, because of the complementarity rule of DNA, they will bind to each end of the DNA, and then they will allow the polymerase, which will happen in the step 3, where we have elongation step. This is where the polymerase start working. Remember, we learned, we took DNA replication, right?
No.
Or you took structure only. we will take DNA replication later, and you will realize that DNA polymerase, the enzyme that makes DNA, doesn't work on its own. It needs primers, okay? And usually in our body, they are RNA primers. But here in a test tube, we are going to synthesize these primers in order to direct the work of the polymerase, in order to make it amplify only a specific region, which is my gene of interest that I want to amplify. Okay? So I repeat, step 1, we heat to separate the double strand. Step 2, we cool so that the primers anneal. Anneal means bind to the right place. And then we have the extension step or the elongation step. This is where the Taq polymerase starts working. So what is Taq polymerase? It is DNA polymerase, which was isolated from the bacteria Taq, Thermus aquaticus. Okay? So they add the enzyme, and the enzyme will amplify that region, and we will start having, so this is one cycle, and then we repeat step 1 to 3 in another cycle. Okay, and we repeat and we repeat. So we have exponential increase in the number of the amplified DNA. That's why we call it a chain reaction. Okay, so it's a chain reaction caused by polymerase trying to amplify region of DNA which has been dictated by the primers. Okay, so as I said, in the test tube we have the DNA that I want to amplify. So if I say I want to see if a patient has sickle cell, so I take this patient DNA, extract the DNA same way that you've done a DNA extraction before, and I put it in a test tube. I put inside the test tube the enzyme polymerase. Okay, what else I need to put in the tube in order to make DNA? So the starting material is with template, the enzyme. What else? Excellent. The building blocks of DNA. What is DNA made from? Nucleotides. So I need to put the nucleotide mix, and this is what we call the DNTP mix. What does DNTP stand for? Yes, so DNTP mix is the nucleotide, the dinucleotide triphosphate. Okay? Deoxyribonucleotide triphosphate. Okay, so this is the 3 major things that you need to do PCR. The primers that will specify which gene you are going to amplify, the enzyme, which is going to do the job of the making of the DNA, and the raw material to make DNA, which is the DNTP mix. Okay? So you mix them with a buffer because every enzyme needs certain pH to work. So you mix them with a buffer and load them, put them in a tube and then put the tube inside the machine, and you program the machine so that you determine the temperatures, and you run it for 30 cycles or 35 cycles, something like that, and you will have an exponential amplification of your gene of interest. Then you can take the sample and load it on a gel in order to visualize it. Clear? Yes?
Yes.
Okay, I hope so. So all of that is written here. This is the kind of figures that you will be dealing with in the exam. So I will be giving you gel and you need to interpret it. Okay, so here we are trying to compare. This is the CFTR. What is the CFTR? Cystic fibrosis. Okay, so we're trying to compare the normal gene to the mutant gene to see if a patient has the disease or is a carrier of the disease. What does homozygous normal mean? Homozygous, what does homozygous mean? Both copies of the gene are identical. Because we have 2 copies of every gene in our body, one from the mother, one from the father. So homozygous normal means both copies are normal, right? Homozygous mutant means both copies have the mutation. And this is the heterozygous mutant. Heterozygous means the 2 copies of the gene are different. So this patient, we call him a carrier. So for CFTR, you need to have 2 copies of the mutation in order to have the disease. If you have only 1 copy, you are called what? Carrier. If you have 2 copies, it means you are affected, so you have the disease. You will study this in greater details in genetics. So what is the difference here in size between the normal and the mutant? What is the difference between?
Mutant is enormous.
Okay, so look at the sizes here. So we have 63 base pair and 60 base pair. So there are 3 base pair. So this is actually the deletion, right? They are 1 codon less. The mutation will lose. There are different types of mutation. One of them is where you lose the 3 base pair. Okay, so this is the mutant one. It has lost the 3 base pair. Okay, so this is an example of application of PCR. So trying to detect whether a patient is a carrier or has the disease of cystic fibrosis by doing a PCR of the cystic fibrosis gene and comparing the size of the gene amplified by PCR to the normal person who doesn't have the disease. Okay? So this is kind of application for PCR, and we will see more applications next week. Make sure you watch the videos, not next week, tomorrow. Okay, so we'll talk about sequencing, other techniques for using DNA, and then move to RNA, then to protein. Do you have any questions before we conclude? Was it all clear? Okay, so I hope you enjoyed Mol 2. By the way, usually students, they enjoy Mol 2 more than Mol 1. And they find it easier. Okay, so usually students find Mol 2 easier than Mol 1. So let's be hopeful, but please work hard.